Hi everyone, my name is Caterina Andrea Ruiz Carp. I am a student from third semester in the Universidad Pontificia Bolivariana, and today I'm going to be presenting to you the investigation of bone mesenchymal stem cell-derived sEV-Encapsulated thermosensitive hydrogels, accelerated osteogenesis and angiogenesis by the release of Exosomal MIR21. So, first we need to understand what is osteogenesis. Osteogenesis refers to the formation or development of bone. This can occur by one of two processes, the intramembranous ossification and the endochondral ossification. The intramembranous ossification is where osteoblasts differentiate directly from mesenchymal cells and begin secreting osteoid. It's the first image we can see here, the A. And the endochondral ossification is where a pre-existing matrix of hyaline cartilage is eroded and invaded by osteoblasts, which then begins the production of osteoid, which would be this image here. What is osteogenesis for? This process is important for the formation, growth, regeneration, and repair of bones. Now we're going to learn what are mesenchymal cells and exosomes. So mesenchymal stem cells are stromal cells that have the ability to self-renew and also exhibit multi-lineage differentiation. So they're pluripotential cells. They can be isolated from a wide variety of tissues, for example, bone marrow, umbilical cord, adipose tissue, blood, and others. And they are easy to harvest, which makes it practical for experimental and clinical applications. However, exosomes are extracellular vesicles generated by all cells that carry nucleic acids, proteins, lipids, and others. They provide cell-to-cell communication by delivering their contents to the recipient cells. They also protect these contents from degradation. A benefit from these exosomes is that they possess stem-like pro-regenerative properties, and they also prevent many of the adverse effects of stem cell transplantation therapy. What's the relationship with osteogenesis? These cells can be harvested from bone marrow and then introduced into an exosome to be administered into a person, enhancing bone regeneration and eliminating the need for alternative procedures. What's the objective of this investigation? The objective is to research the properties of bone mesenchymal stem cell-derived sEVs encapsulated in thermosensitive hydrogels for the acceleration of osteogenesis and angiogenesis by the release of exosomal MIR21. Now we're going to talk about some of the methods utilized in this investigation. First of all, we have the Western blot, which is a similar technique to the Southern blot used for the detection and identification of specific proteins. In this method, the proteins are separated depending on their size through porogel electrophoresis, as you can observe here. Then, the proteins are transferred and incubated with an antibody that reacts to the protein of interest. So this process right here. Then this antibody can be detected in various ways, allowing the protein in which the antibody acted to be discovered. So we would finally see it like this. Now, the RT-qPCR. With this technique, it is possible to determine the expression of genes in diverse tissues, and it increases the reliability of gene expression analysis. So what we're going to do with this test is amplify the genetic sequence, the genetic material. Why was this test utilized in this investigation? Because it allows us to find the range of expression of the genes in the mesenchymal cells that would carry out the function of osteogenesis and angiogenesis. Now continuing with other methods, we're going to talk about immunohistochemistry. It's the identification of a tissue in situ by means of a specific interaction between an antigen and an antibody, monoclonal as well as polyclonal. So the antibody has been visibly marked, and also the cell is colored to demonstrate the presence and cell location of a molecule and interest within the protein. Next, we have the immunofluorescence test, which is a type of immunohistochemistry technique that utilizes fluorophores. This would allow us to visualize various cellular antigens such as proteins. This technique can be utilized to visualize the location of various cellular components within cells. Now, going on with the results of this investigation, we can see in the first image, image C, that it's a Western blot analysis to compare the protein expressions between normal cells and the BMSC sEV cells. What is a BMSC? It's a bone mesenchymal stem cell. In this case, it has a sEV introduced into it. So we can see that the modified cell expresses CD81, CD63, and TSG101, but it doesn't express count-nexin. Also, we can see that in the modified cell, the bands have a darker coloration and a thicker width, which means that the protein expression is higher in these types of cells. Now, passing on to the next image, image D, this image is a immunofluorescent test done to compare the expression between the BMSC cells and the Huvix cells. What is a Huvix cell? It's a human umbilical vein endothelial cell. So we can see that the cells were dyed with 3 different colors, well, with 3 different dyes. You can tell by looking at this image that the BMSC cells have a higher expression because their coloration is stronger in comparison with the Huvix cells. And this test showed that the sEVs could be observed in the cytoplasm, which was indicative of their attachment and internalization by the BMSCs and Huvix. Next, we have other tests. Image D and image F are both Western blot tests, which are testing the protein expression in different concentrations. And as we can see, both of them have a test control in this case, the GAPDH. Why? Because every cell expresses this. Now, specifically image D. In image D, the modified cells, the BMSC cells, can upregulate the expression of osteogenic genes. Also, we can see that at a higher concentration, there's even more even more protein expression because the bands are darker and thicker. Now, the image F, so the same thing happens with the first Western blot. At higher concentrations, there is a higher protein expression. Now, continuing with image A, the image A is a Transwell assay and quantitative analysis. So, the Transwell assay measures the capability of a cell to migrate towards a chemoattractant. In this test, we can see that the migratory capability of the Huvix exposed to sEVs was greater than in control cells, and the effect were dose dependent. Continuing with the discussion, a lot of these comments from other authors are true in comparison with this investigation. And now for my personal conclusion. I consider that molecular biology holds a very important place in the medical field because without it and the different processes that it's allowed us to uncover over the years, we wouldn't be able to develop medicines, vaccines, special treatment plans, and others. Also, this investigation nowadays represents an enormous breakthrough that opens the door for the medical field because it can possibly allow patients to be treated with sEVs containing these hydrogels, inducing angiogenesis and osteogenesis. And it would allow us to avoid invasive or more elaborate procedures